I'm going to move on now again to the next page. So I think it's Yuki. And Yuki is going to tell us about LPIXEL. LPIXEL is a really interesting image analysis technology, which is they've been working LPIXEL, they've had a focus on the pharmaceutical industry. However, I think it could be very interesting as well when we're looking at bringing in technologies from pharmaceutical industry and applying them to our research and development processes in agriculture. So I'm going to give you the floor, Yuki, straight away for you to tell us much more about LPIXEL. Thank you very much.
Okay, thank you for your introduction. Okay, hello everyone. Thank you for having us this wonderful opportunity. I'm honored to be here. My name is Yuki Shimahara, COO and founder of LPIXEL Inc. Today, I'd like to talk about ImaCell, which is cell imaging technology. First, let me explain our company. LPIXEL is a startup company from research lab of Tokyo University, which is a pioneer of bioimage informatics. Over 60 people are working in LPIXEL. And one unique point of LPIXEL is that we have invested by large corporations in the life science industry, like Canon, Olympus, Fujifilm, and so on. So we are the hub of open innovation and push for new business opportunities. Our strength is that we have many members who are familiar with both life science domain knowledge and AI. Pure AI engineers are not good enough for research discussion with biologists. We, as a biologist, discuss everything from cell culture conditions, standardization of imaging conditions, and provide the optimal software. Today, I'd like to introduce an example of AI in drug discovery, which are working with pharmaceutical companies. In general, several experiments are used to evaluate target compounds. The evaluation of phenotype is often subjective and monotonous, and it is a huge amount of work. However, with AI, it is possible to achieve high throughput. For example, we quantitatively analyze each cell from a large number of images to speed up the analysis. And by utilizing deep learning, we can capture features that could not be analyzed before. We are currently working on this theme with Daiichi Sankyo and Astellas Pharma, the top 3 pharmaceutical companies in Japan. ImaCell is currently being used by many pharmaceutical companies as an AI that accelerates drug discovery research. By all means, we would appreciate it if you could discuss what we can help with using our technology. Thank you for your attention.
Thank you so much, Yuki, for introducing your technology, your imaging technology. And I think it's always interesting because these cross technologies which are used in one industry and brought to another, these can lead to very exciting developments. And already there's a question come in from Giuliano. And so Giuliano's interested to find out, so have you ever worked with fungal pathogen cells? And the second part of the question is, do you think that your imaging technology could be used with fungal pathogen cells?
Okay, yeah, of course. So I can say that we can analyze all images, so taken by microscope or camera or anything. Yeah, but analyzing that kind of life science images, we need to use the life science domain knowledge. So that's why we can say that we can develop best solution for them. Thank you for
Yes is the answer, Giuliano. That's an interesting one. In terms of images to train the model, so how long in time does it take you to train your model? And do you have an idea of how many images you need to train your model?
That's a great question. Yeah. So I can say that it depends. Yeah. Yeah, so that's why so POC, so small start is the good start. I can say that. So we can, yeah, of course, the more images, the better to develop good images. But for example, we can start about few hundreds images and then do POC, and then so as that result we can see the final output. So if you can achieve 60% using a few hundreds, and then we can aim that the 80% using 1,000, or 95% using 10,000 images, like that. So that communication is a must to start, I think. Thank you.
Thank you, Yuki. And then a last question here is it possible to integrate your technology into another bioimaging platform, or do you have APIs to make that happen?
Yep. Great questions. Yep. Actually, we have the ImageJ plugin as well. So in our ImageJ plugin is the number of install is over 30,000. I think it is one of the best plugin we developed in the research lab. Yeah, so yeah, of course we can provide that plugins, and also if needed, so we can help the customize or the discussion or like that.
Thank you so much, Yuki, for taking the time today to pitch, and thank you so much for answering the great questions from the Q&A.